Good morning, Honorable Person Chowbak here. I am N. Gomati, Assistant Professor of Biomedical Engineering, Erode Engineering College. Now we are discussing about the subject name is biosignal processing. First, biosignal processing. First, you need one title is Introduction to Biomedical Signals. First one, the portion of the syllabus of biosignal processing. Unit 1 Introduction to Biomedical Signals topic is biosignal characteristics of electrocardiogram, electroencephalogram, electromyogram EMG, electroocculogram EOG, electroretinogram ERG, electrogastrogram EGG, electroneurogram ENG, event-related potentials, phonocardiogram, speech signal, objectives of biomedical signal analysis, difficulties in biomedical signal analysis, computer-aided diagnosis. First one, main thing of signal versus the biosignal. What is the difference between signal and biosignal means? The signal is a function that conveys the information about one person to another person or transmitter to receiver. This is called a signal. Carrying the information, this is called a signal. But biosignal means the living, any signal in living beings that can be continuously measured and monitoring is called as biosignal. This is the main difference of the signal and biosignal. The bioelectric signals are low level amplitude and frequency range are obtained. Next, now types of biosignals. What are the types of available in biomedical signals? Are bioelectric signals means electrocardiogram, electroencephalogram, electromyogram, electroretinogram, electrooculogram, electroneurogram, electrogastrogram, phonocardiogram, event-related potentials, speech signals are included the sessions. First one is biosignal processing. Biosignal means the taking the bioelectric signals from the electrodes are used to taking the bioelectric signals in human body. But biosignal processing means the signals are extracting significant information from biomedical signals. This is the aim of the biosignal processing. With the aid of biomedical signal processing, biologists can discover new biology and physicians can monitor the illness, measure the amplitude and frequency levels and the ability or disorder or enable or normal or abnormal conditions of electrical activity of human being. Electrical signals are measured and monitoring of biosignal processing method is used. First one is ECG, electrocardiogram. Electrocardiogram means the electrical activity of the brain, contractile activity of the manifestation of the contractile electrocardiogram means a contractile activity of the heart and can be recorded fairly easily with the surface electrode on the limbs or chest. The rhythm of the heart in terms of beats per minute, BPM, beats per minute. The normal heart rate is 72 per minute. Beats per minute is normal human being heart rate is 72 beats per minute. Next is the more important is the fact that the ECG wave shape is altered by cardiovascular disease or abnormalities such as myocardial ischemic ischemia and infarction, ventricular hypertrophy and conduction problems. Next, heart. Heart is four-chambered pump with the two atria and two atria for collection of blood and two ventricles is for pumping out of blood. This is called as co-chamber of heart. The resting or filling phase of a cardiac chamber is called diastole. Pumping phase is called systole. This is a heart structure of the diagram. This diagram, the heart is very important. 4 chambers: one, right ventricle, right atrium, right ventricle, and left ventricle. Next, left bundle branch, Burgund's fibers, this bundle, so many parts, so many works are done by heart. The 4 chambers available. The impure blood from the right atrium is collected from the impure blood from the from all our bodies. Uh, but uh, uh, but right ventricle is the impure blood is given to the lungs. The lungs is segregate by impure blood into the pure blood. This this way of the right ventricle used. The left ventricle or left atrium, left ventricle received the blood from lungs, and left atrium distribute the pure blood in all parts of the body. This is the main thing of the heart functions. Next, heart rate or cardiac rhythm is controlled by specialized pacemakers selected from the sinoatrial node, SA node, and ANS leading systems. And normal heart rate is 70 beats per second minute. The heart rate is lower during sleep, below 60 beats per minute. Bradycardia, above 200 beats per minute is dicardia. Conduction system of the heart. Conduction system is the cardiac cycle as follows: SA nodes. The P wave is generated in the EC waveform, and the low P wave is low amplitude wave. The amplitude about 0.1 volt to 0.2 millivolt. The time duration is 60 to 80 millisecond. This is AV. Next one is PQ segment. They generate the AV node. Next is QRS of the ECG wave. This segment is called as ST segment. The ST segment amplitude range is 0.1 to 0.3 millivolt, and time duration is 120 to 160 milliseconds. This is the waveform of the action potential curve. ECG waveform of PQ or ST levels. The time duration is given to the diagram. The time duration is each and every PQ or ST. The time distance is 80 millisecond. Time interval is 80 millisecond, and S and T the time interval is 100 millisecond, and T time interval is 160 millisecond. Overall is a Q or ST time interval is 340 millisecond. The action potential of atrial myocard and action potential of ventricular myocard diagram is shown in figure. This is the cardiac cycle. This is already SA node and AV node and ST node are also explained in. P means atrial contraction, PQ means segment isolectric AV delay, and QRS means ventricular depolarization, contraction or systole. This is a ST segment. ST segment is isoelectric and plateau of action potential of ventricular myocycle. T means T interval means ventricular repolarization and ventricular relaxation are diastole up to next beat. This is called as cardiac cycle. The routine of PQ or ST cycle is next beat level is automatically generated in the ECG waveforms. The SA nodes, AV nodes, and the ST nodes are noted. Arrhythmia. Arrhythmia means the disturbance of the disturbance in the regular rhythmic activity of the heart is called arrhythmia. SA node is normal or SA node is irregular firing patterns from the SA node, and the QRS shape is affected by conduction disorders. This is called as arrhythmia. Next one, ECG lead system. ECG lead system consists of two surface electrodes of opposite polarity or one positive surface electrode and a reference point. The lead composed of two electrodes of opposite polarity is called as bipolar lead. A lead composed of a single positive electrode and a reference point is a unipolar lead. This is a diagram of the 12 lead system 12 lead system ECG lead system diagram. The 12 lead system ECG consists of three bipolar limb leads as the limb leads 1, limb leads 2, and limb leads 3. And the unipolar limb leads are AVR, augmented vector right, augmented vector left, and augmented vector foot. And 6 unipolar chest leads with V1 to V6 are used as the precordial or V leads are used in ECG lead systems. The first A diagram is shown in ECG consists of bipolar lead system, lead 1 system. The right arm is negative and left arm is plus connected to the electrodes electrodes used to measure in lead 1 system. Lead 2 system is right arm is negative and chest is positive polarity is connected to lead 2 systems is measured, and the left arm is negative and chest is plus. The third C diagram is noted in lead 3 system measured. The last one is the diagram is shown in figure. This is called as enthoven triangle. The right arm and left leg are used to triangle shape of the lead systems. The lead 1 and 2 and 3. These systems is also called as enthoven triangle. Next again lead 1, lead 2, lead 3. This lead 1 is right arm negative, left arm positive. The records electrical difference between the light and the right arm electrodes, left and the right arm electrodes. The lead system 2 is right arm negative, left leg positive. Left leg positive is a records electrical difference between the left leg and right arm electrodes. Lead 3 means left arm negative, left leg positive, records electrical difference between the left leg and left arm electrodes. Next to the lead system of unipolar lead system of lead AVR means augmented vector right, positive electrode right shoulder. So diagram is shown in figure. First diagram is AVR, augmented vector right, and augmented vector left is positive electrode, left shoulder. The AVF means augmented vector foot, positive electrode on foot. This is chest leads of the first one. Lead 1, 2, and 3 are bipolar leads, and unipolar 3 leads are used. 6 plus chest leads are 6. V1 to V1, V2, V3, V4, V5, V6. Chest leads are insert in the electrodes. The lead system chest leads are measured. The 12 lead systems are obtained in ECG lead systems. This is the diagram of the ECG waveforms 1, 2, 3. Is the first one is lead 1, 2, 3, and AVR means augmented vector right and augmented vector left and augmented vector foot, and chest leads are V1, V2, V3, V4, V5, V6 waveforms are shown in figure. Next topic is electromyography. Electromyography means the electrical activity is the response. The measures of muscles response are electrical activity in response to your nerve stimulation of the muscles. The test is used to help detect neuromuscular abnormalities. During the test, one or small leaders, the maximum small lead electrodes are used to measure the electromyograph. The conduction velocity between the EMG waveform is 50 to 60 meters per second is considered in normal position. Below 50 or above 60 conditions is conduction velocity is the abnormal of the muscles and muscles. This is the EMG recording systems. EMG recording system, EMG surface electrodes are placed in this format first diagram. The second diagram is EMG electromyograph waveform is noted. First one is the amplitude level is 50 microvolt. The time period of contraction relaxation time difference is 1 second. Contraction means the expanding of the signal, electromyographing signal. Relaxation means contraction means compress of the signals. Relaxation means expanding of the signals in electromyograph signals. Up to the frequency level is 1 kHz, up to amplitude level is 3 millivolt, up to 3 millivolt in EMG signal. This is the recording setups and the waveform of the EMG. Next, EMG waveform. The interference pattern. First one is interference patterns available. First diagram is the EMG electromyograph, the normal trace. Second diagram is neuropathic trace and myopathic trace waveform are obtained in EMG recording systems. Next one is the electroencephalogram. The electroencephalogram that it detects the abnormalities in your brain waves or electrical activity of the brain are measured. The electrodes are consisting of small metal disc with thin wires are pasted onto your skull. The electrodes detect tiny electrical charges that result from the activity of your brain cells. The EEG can help diagnose a number of conditions including epilepsy, sleep disorders and brain tumors or diagnosis purpose use of EEG. Epilepsy means the fixation. Many people are affecting fixed level. This is called a fixed level is called as epilepsy. Sleep disorders and brain tumors, brain disease or measured is identified the brain tumors and diagnose the problems. These are rectified in EEG recording systems. The electrodes are used to electrodes 10 to 20 systems electrodes are used. Placement is a method used to describe the location of the scalp electrodes. The record is the result of the activity of thousands of neurons in the brain. The 10 to 20 systems diagram is shown in figure. The four loops, central loops, 4 loops are used in the placement of electrodes in the scalp. The letters of F means frontal loop and T means temporal loop, C means central loop and parietal loop, P means parietal loop, O means hospital loops are used to measure the placement of the electrodes. Even number of 2, 4, 6, 8 refers to the right hemisphere and odd number is 1, 3, 5, 7 is left hemisphere. That refers to the electrode placed on the midline. Nasin means point between the forehead, and nose, point between the iron is pumped back of skull. This is a spacement of the electrodes in electroencephalogram signals, EEG signals. The electrode phase in the frontal loop is F1, odd numbers in left hemisphere and even number is right hemisphere. The nasin is the forehead between the nose, forehead and between the forehead and nose. Nasin. This inion is back of the skull. This is a diagram placement of the electrode placement of the EEG 10 to 20 lead systems. This is a waveform of the EEG waveform. The EEG waveforms, depending upon the human being thinking level or sleeping level or concentration or drowsiness and sleep and dreaming, any other thinking, whatever it may be, the brain's waves are depending upon the body, depending upon the resting or action potential. The waveforms are changed in brain waves. First one is gamma. Is a problem solving concentration is the gamma waves. First diagram. The beta is a busy or acting mind of people's. The waveform is measured. Alpha means reflective, restful mind is the brain waves is measured. Theta is drowsiness people's waveform. Delta is sleep and dreaming waveform. This next one is EOG means electroocculogram. Oculography is a technique measuring the corneoretinal standing potential that exists between the front and back of the human eye. This diagram is the electrodes are placed in the one electrodes are placed in the forehead and another two are placed in the size of the eyes of the end. Silver chloride electrodes are used in the electrooculogram measuring purpose. Next one is EOG waveform. First one is looking straight ahead. Second one is rolling eyes upward and rolling eyes downward is third diagram. This 1 and 2, 3 diagrams are used to measure the waveform of the electrooculogram. First one is original first waveform x-axis original EOG waveforms time millisecond and EOG waveform signal of amplitude levels y-axis. The amplitude levels first one is looking straight ahead means the amplitude levels and frequency level is very low is compared to two diagram and three diagram. First rolling of eyes upward and downward, the amplitude level is changed and up and down changed. The amplitude levels and time frequency level changed on the waveform of EOG. Next one is ERG, electroretinogram. Electroretinogram means electrical retinogram is a diagnostic test that measures the electrical activity of the retina in response to a light stimulus. ERG arises from currents generated directly by retinal neurons in combination with the contributions from retinal glia. Electroretinography waveform is shown in figure. First one is A wave and B wave and baseline are used in ERG waveform, electroretinogram waveform. The baseline is A wave latency. Is the A wave input time is A wave latency. The input time is short and then increased the B wave implicit time is increased. The B wave amplitude and B wave implicit time is continuously increased. The B wave is diagram is suddenly particular time difference. The B wave is increased. Suddenly decreased the baseline of electroretinogram waveforms. So B wave indicates the activity of the amacrine horizontal bipole and Miller's cells. The peak levels of A and B waves of initiation of the light stimulation cycles. A wave amplitude was measured from baseline to a wave through. B wave amplitude was measured from A wave to B wave peak. This is ERG, electroretinogram waveform. Next one is electroneurogram. Electroneurogram method is used to visualize directly recorded electrical activity of neurons in the central nerve systems or the peripheral nerve systems. ENG, electroneurogram recording system. A concentric needle electrodes are silver chloride electrodes under the surface of the body. They mainly used to measure the nerve conduction velocity. This is a electroneurogram diagram, the electroneurogram conduction velocity is formalized distance divided by latency 1 minus latency 2. What is meant by distance? The distance between the distance stimulation and the proximal stimulation distance is also called as distance. The recording of the electrodes are placed in the palm. The two electrodes are used to measure the ENG electroneurogram. The distance is one point to another point is the distance between the distal stimulation or proximal stimulation. The latency one diagram is two waveforms are used to do the latency 1 and latency 2. The time difference between the distance is called as latency 1 and latency 2. The conduction velocity formula is calculated by distance divided by latency 1 minus latency 2. This is called as electroneurogram. Conduction velocity measured by waveform. Next one is electrocardiogram. Electrocardiogram is here. EGG, electrogastrogram is a painless test that detects problems with the nerves and muscles of the stomach. The electrical signals are to measure the activity of muscles and nerves in the stomach. An EGG can help diagnose stomach symptoms including gas and chronic nausea, vomiting and indigestion. This is the surface electrogastrogram waveform. First one is waveform of normal and padiagastria and dicastria waveform. First one, second diagram is surface electrode used to measure the electrical activity of the muscles and muscles and nerves of the stomach of our body. The electrodes are placed in the 1, 2, 3. Red color and yellow, black color wires are used to electrode. Surface electrode are used to pace the abdomen. The electrical activity of the muscles measured and waveform is used to recognize the normal and padiagastria. Normal means 1000 microvolt amplitude levels and 60-second time period. But bradycardia means 500 microvolt only. 500 microvolt. Tachycardia means 500 microvolt. This is normal. First one diagram is normal EGG, electrogastrogram diagram. Bradycardia and tachycardia. Second and third diagram is shown in figure. Next one is phonocardiogram. Phonocardiogram means is a plant of high fidelity recording of the sounds and murmurs made by the heart with the help of the machine called the phonocardiograph. The phonocardiography is the recording of all sounds made by the heart during a cardiac cycle. Cardiac cycle means PQRST levels. This is called as cardiac cycle. Next one is PNG, phonocardiogram. Waveform. First one is first, second, third, and adrenal waveform. I used it. A, B, C, D, E, F. Diastole, systole, diastole, systole. This waveform is changed in diastole and systole. First one is normal. First diagram is first, second, third. Adrenal is a normal. Next one, the aortic stenosis, mitral regurgitation, aortic regurgitation, mitral stenosis, ventricular septal defect. These are the types of the phonocardiogram waveform, phononeurogram waveform. This is a phonocardiogram instrument. This is a measurement of the phonocardiogram. This is a components of the PDA based digital phonocardiographic equipments. Next, event-related potentials. Event-related potentials means formerly termed evoked potential or event-related voltage changes in the ongoing EEG activity that are time-locked or to sensory, motor, and cognitive events. Event-related potential to identify and classify the perceptual memory and linguistic operations. It is very small voltages generated in the brain structure in the response to specific events or stimuli. This is the conclusion of the first unit of biosignal processing unit. Is a conclusion. This is the conclusion of biosignal processing. First unit is how to make or monitoring and measured of the bioelectric signals are measured. Where are the any kind of electrodes are used in measuring of the electrocardiogram, electroencephalogram, electrokilogram, electrogastrogram. So many bioelectric signals are measured and monitoring, and normal or abnormal conditions are identified and problems are rectified in biomedical systems. Biomedical systems. Thank you. Thank you.